All right. So first of all, I apologize for not being there in person. I had a medical issue with the family that kept me from coming. So I regret that very much. And so I tried to stay in my time here. My name is Ulrich Reimann-Philipp, and I run the tissue culture lab at Tent Harvest Cultivate in Grand Rapids, Michigan. So what I want to talk about is how we use tissue culture to maintain our genetic assets and how we manage the hop latent viroid epidemic that has really caused a lot of damage. And it's a big concern here for cultivators in US and Canada. And I don't know how it is in Europe yet. If you haven't had a problem with it yet, you definitely will pretty soon. So I'm working for Tent Harvest Cultivate, or THC. We were called Flourish before. We are one of the top 3 cannabis cultivators in Michigan. We operate 2 facilities, one indoor facility here in Grand Rapids and a greenhouse facility in Adrian that you can see in the background there. Combined, they have about 8,600 square meters of flower space, and we're producing, our biggest business is wholesale flower, so we produce about 450 kilograms of dried and trimmed flower per week. And we also have extraction manufacturing laboratories to produce concentrates and pre-rolls and gummies and so on. So this might be familiar, or probably is familiar to most of you, but I still like to repeat some basic facts that apply to what I'm talking about here. So of course, all the cannabis production plants in the marijuana side are propagated through cuttings from so-called mother plants in a process called cloning. So these clones are genetically identical to the mother plant, like identical twins. And that is important because one of the reasons is only female plants can be grown for production. Any male plant in your cultivation room or greenhouse spoils the whole harvest because you would have seeds instead of flowers produced. Also, seed propagation is not very suitable for what we're doing because when you germinate seeds, you get female and male plants, and they're like brothers and sisters, so genetically much more variable. That means that all big growers have their own stable of mother plants that represent their genetic portfolio of the different strains or cultivars they have. And this is like a very important asset because once you lose your last mother from any kind of premium strain, you lose the whole strain. So we started our tissue culture program about 3 years ago. I came from California at that time when THC was just opening here. And it is basically a method of propagating plants from very small pieces that we call explants and do that in a sterile environment. The advantage of it is that you don't transmit a lot of the diseases and pests from the mother to the clone. So if you have a mother that is infected with something, or it has russet mites or other kind of diseases or pests, then it would be passed on to the clones. So with the tissue culture, you can produce healthy mother plants that are free of all the pests and pretty much all the diseases, except for some virus diseases that are not easily removed. But that is a way of preserving your genetic assets. And we use the tissue culture to maintain a stock of mother plants. And we also take advantage of the fact that through tissue culture, you can often get rid of epigenetic changes. So these are basically changes that happen without actually changes in the DNA sequence, but that can occur over time if you copy and copy and copy through cloning. So we also have some research projects that we're working on. Just mentioned this briefly. So one is cryopreservation of genotypes like a sperm bank. So we're just right now developing this and keeping them in liquid nitrogen for long-term storage. And then, of course, at some point, tissue culture is will be essential for cannabis and genetic engineering, and we work on this as well, but that's still in the very early stages. Here's a picture of our tissue culture lab, or part of it. We operate these two laminar flow hoods, and the rest of the lab is not on here, but we also have the capacity to climatize our cultures, to run them at higher temperatures or lower temperatures, depending on what we want to achieve with that culture. And we're operating it with a team of 3, 2 techs and a manager, and then myself is overseeing the whole operation. This is how it works. So we start with a mother plant. I don't know, can you see that cursor that I have on here? If not, then the mother plant is on the left. And the first thing we do is we cut very little pieces of that mother plant. So all of these contain what's called a meristem. It's kind of like a little cluster of stem cells. And these pieces are usually like 5 millimeters tall, and we can sterilize them and then start a culture in a sterile environment on sterile media, like a solidified nutrient and plant growth regulator mix. And you see that in this box up there. So what we do is we put these little explants on the induction media, and then we have little shoots growing out of it, which when they have a certain size can be rooted either still within this agar media, or they can be transitioned straight to rock wool or peat wool to root. The one thing that we're still working on, we haven't had any success with, is going through stage called callus, where this explant grows into some kind of non-differentiated glob of cells. If you induce this in the right way with the right kind of growth regulators, you can get a lot of shoots out of that all originate from a single cell. It's called somatic embryogenesis. And then you have a lot more offspring from the one explant. And it's very much used in other plants, especially in a lot of ornamentals and in berries and fruits. And so that's what you can do to produce a lot of offspring in a small room. And that is not really working for cannabis yet. So once you have your little tissue culture clones rooted, they grow up into mother plants, just like a conventional clone that you see on the bottom there. Here's a close-up of the explants that we're starting. So this is not just the meristem, it's some tissue around it and some leaf primordia, and they're very small, and the smaller they are, the better your chances is to exclude any kind of diseases or pests that might be in the mother plant. Then it looks like this. That's how it starts. So we have these little explants on the cubes and on the agar media, and then they will start growing. After about 2 weeks, they look like this. So they already start looking like a little plant, and eventually they're big enough that they have a stem and leaves, and they start making photosynthesis, and you can cut those and root them, in this case, directly on little peat cubes. So this is how it starts. These plants look different from regular clones, which are a lot bigger, and also from seedlings, so they don't have any cotyledons also. They're basically just like tiny little mini shoots. And then eventually when they grow up, you cannot distinguish them from regular clones. They look just like this. This is a view of one of our mother rooms. So they grow that big. We usually replace them every 3 months. We don't want the mothers to get too big, because then they are more likely to catch any kind of diseases or pests. Now, the big reason why I was brought here to start this tissue culture program was that hop latent viroid started to become a major problem in the industry here in Michigan, but also everywhere else. It's kind of like the COVID for cannabis. It causes what's called the dudding disease. It was first reported in California, and now is present in about 90% of the California operations, and the 10% that has not been found yet will probably get it at some point or another. And it affects, so far as known, 2/3 of the US cultivation facilities, and it keeps spreading. The annual crop losses here in our industry are estimated like about $3 to $4 billion, depending on the way they are calculated. The disease is caused by a viroid. It's called hop latent viroid. So it came from hops originally. It's pretty much everywhere there, but since it's a latent, as it says in the name, latent viroid, it doesn't cause a lot of symptoms and doesn't cause a lot of damage in hops. So it's a very unique type of pathogen. It's like just a little ring of RNA, about 256 bases long. It doesn't code for anything. It doesn't have a protein code like viruses has. And once it gets into a plant cell, this cell will produce copies of this viroid. So millions of copies, and eventually that affects the performance of the plant. And the symptoms and how HLV is distributed and transported within and between plants depends on the mode of infection, the age of the plant, and likely all kinds of environmental triggers. For sure, light period and certain stresses can switch a latent infection to one that causes severe symptoms. Now, one problem with the HLVD infections is that often you don't see any symptoms in the early stages. So the cannabis is grown from clones, and then it will be maintained for a while in a long light environment where it won't flower. And this is the vegetative stage. And many times you don't see any symptoms at that stage. Only when it flowers, most of these become apparent. And they're usually just smaller plants, so there's some stunting. The branches and stems can be very brittle and break easily. They also branch more horizontally, and I'll show you a picture of that later. The flowers are not as dense, or the buds are not as dense, and you have reduced yield and potency of these plants. Sometimes you also have symptoms on the leaves, but not always. Now, the reduction of even 20% of your yield and potency for an industry like ours here is make or break, because the margins in the cultivation are small, and the prices have come down a lot. So we cannot really afford to lose any of the potency and yield that we usually get. Here's a picture of two mother plants, one infected, one not infected. Both came from the same cloning batch, and other than the fact that one is a little bit smaller than the other one, you can't really see any symptoms. So the leaves look all good, the plants look healthy, you can take clones from them. Usually the ones from the HLV infected moms don't root as well, but other than that, since both of these plants have been cloned from several times, you can't really see the symptoms. Once you switch into flower like these here, you do these plants up front, the HLV plus plants, they are infected, and they just are smaller, they're kind of brittle, they don't grow as high, they are not as dense, and those plants are really pretty much a loss for your harvest. If you look at the branching, that is this picture on the right side, you see the infected plants, where you see the branches come off the stem almost horizontally, whereas usually they're like about a 45-degree angle. There's still some questions about how HLV is transcribed. The most common one is definitely cloning from infected mothers. So if the mother has it and you clone from it, then your offspring will have it. And that is to a large extent also true to tissue culture and micropropagation. If the mother is thoroughly infected, every part of the mother has it, then your offspring will have it too. So there are certain ways you can treat those cultures to get rid of the virus, but tissue culture alone does not remove the virus. Very efficient is also transmission by plant saps. So that's mostly through cutting and pruning tools and de-leafing. If there's not the proper hygiene between plants, if you take the same snippers to cut from one plant and then go to the next, that's a very efficient way of transmitting HLVD. It also can be transmitted through seeds. So if you buy seeds from somewhere or you break your own, you can assume that this infection will spread through the seeds just as well. And we have seen it often in our seeds that we have germinated here. There's also transmission through water that has been shown in the lab quite convincingly, but I don't know how big a problem that is in large-scale cultivation. Definitely when you have a hydroponic system that recycles the water, then you're very much susceptible to that kind of transmission. And insect and fungal vectors are also suspected and very likely can carry this viroid, but there's no experiment yet that shows that conclusively. Now, how do you detect HLVD in plants that don't really show any symptoms? That is really, there's not much you can do with these vegging plants. If they don't grow symptoms, you don't know. And if they do have, then these might be having different causes as well. So you can have stunted plants for many different reasons other than hop latent viroid. So the only reliable way at this point is reverse transcription qPCR. So that's called a real-time PCR, where you actually detect the RNA, the viral RNA itself. It's very reliable, it's very specific, but comes with some cost. So it's basically the same type of test that has been done for COVID testing. So we have our own in-house qPCR testing. There is an upfront cost for equipment. So the PCR machine and some of the lab equipment we bought cost about $35,000. And then the reagents per test are about $9 to $10 if you do it exactly as the kit would prescribe. It's still a lot cheaper than buying your test or having your test done by an outside lab. You have a same day turnaround, so you can immediately see what your results are. The test data are much more reliable than from a test lab, and that is, if you see in the background these curves, that are fluorescent curves, that's kind of the results that you get with this qPCR. So you're creating a fluorescent through the consecutive amplification cycles of that RNA. And there are sometimes curves that don't look that clear cut, that might be a little bit having an odd form. And usually you get the results from a testing lab back as a CQ value, which is basically the point, the number of cycles of amplification where your curve crosses this line that you see up there, your baseline. But the baseline can be set arbitrarily. So you can have false positives or false negatives, and depending on where you set this line. So you can reduce the cost if you have some experience with using smaller volumes and sourcing of the reagents and so on. That's what we do, and our tests are more like $3 or $4 a test. And the equipment that you can use also for other tests that are useful in your grow, that is definitely for gender. If you germinate seeds for pheno hunting, you can identify the males very quickly and throw those out. You can test for botrytis and fusarium and other fungal pathogens, other viruses, then not just hop latent viroid, but other viruses as well, and things like russet mites that sometimes are hard to detect. Important when you sample for HLVD testing is that you collect more than one sample that you then combine in one reaction. And this is because HLVD is not evenly distributed to the plants. Sometimes you have plants that have HLVD in some parts and not in others. So if you only test like a single leaf, you'll easily miss a positive plant. And you can either sample the roots or the leaves, but in each case, you have to combine several samples to get reliable results. So our strategy for HLBD control is that we maintain a stock of all our strains in the form of mother plants that are produced through tissue culture. So every mother plant that we have in our mother rooms came from a tissue culture propagation, and that comes up to a library of stock moms, if we call them, or master moms. So we maintain one or two of each of our strains. And then all these moms must test HLBD negative before they move from the tissue lab into the cultivation rooms. And once they're there, they are being tested monthly. That is important because you can always get this infection coming back in with your personnel or with some insect vector or with something that we don't even know about yet. So also it's important to have proper sanitation procedures, meaning that gloves are being changed before working on different plants, or that also that all your tools have to be sanitized in between plants. So usually we use 10% bleach to soak our tools, and that has shown to be a very efficient way of preventing transmission. We also have all our pots and rock wool blocks on these little razors, so they're basically preventing any kind of runoff that comes from one block to have contact with the other plants. So in summary, I just want to stress that HLBD is here to stay. It's going to be there. It's hard to detect, definitely in vegging plants, often not at all. It can come into your facility through different channels, and you can never really be sure that you get rid of it. Even if, like we have it here sometimes, that every sample at some cycle of testing tests negative, and then you'll test again in 1 month, and several of the plants might show up positive. So it's something that is an ongoing project. The key battleground for your HLBD control is your mother room. That is most important to keep this mother stock healthy. So one thing is to produce your moms through tissue culture. That helps, and to test all these moms regularly and recycle them at least 3 times yearly so they don't get too big. The bigger they are, the more likely is that something creeps in there, not just HLBD, but all kinds of other fungal and other infections. So if you keep a library of these stock moms and you keep them healthy and recycle them, you cannot lose then you won't use your strains to any kind of virus or other diseases. And never take clones from vegging plants for the next grow batch. That's what some cultivators here have been doing. So basically they take the tips of one batch and use it to start the next one. That's a sure recipe for disaster in one way or another. It doesn't even have to be HLBD. Then, of course, it's important to enforce all the sanitation protocols. And you can assume that any plant material that you bring into your facility from outside sources might be infected. And we have seen this again and again, being it whole plants or seeds or clones. You really need to quarantine them and test them several times before you trust them to be part of your operation. So thank you very much for listening, for letting me present this here, not being able to come, and I appreciate your attention.